Welcome back. Today, I'm going to show you how we can use densitometric analysis if you're working with Western blot or, for example, some of the dot blot type of experiments. Just briefly, these experiments are targeting the protein expression. So with the antibody detection, they were using most of the cases X-ray films, and that is going to give you the result in which the black dots or the black bands will be there. Nowadays, more frequently, people are using the digital version of the developing method, in which you would get the same results, but in that sense, they are using computer and a special camera to record the expression of these proteins. But the end, it will be exactly the same. You would get different bands and different dots. So today, I'm going to show you how you can use the so-called ImageG program. And this application is not only limited to densitometric analysis, but also it can work on, for example, if you're working with bacterial culture and you need to count the dots on the Petri dish, or even if you are working with microscope slides and you need to determine how many cells are there on the screen, it is also an option as a plugin in this program. Today we are going to focus on the densitometric analysis. So let's jump in and let's see what we can do. First of all, if you are opening the ImageG program, the basic setup that it is on the rectangle type of selection, but you need to open the file first. So I did previously an image in which I put all my protein expression images together, which helps me to analyze the data this time. So I'm going to click on File and I'm going to open it. In my case, it is on the desktop, so I'm going to open that figure. This is how it looks like, and just to make super clear, these are theoretic protein expressions, so that's why I just use the blah blah type of terms in order to avoid any kind of identification. These are my older results, but that's good for practice here. Also, as you see, I was marking 3 different types of proteins and also loading control. In the case of the Western blot, the loading control is the super crucial part, because this is a protein expression which does not change upon any kind of treatment. People usually using GapDH, lamin, actin type of expressions, because these are the ones which are not going to change upon the treatment that they are using. And if these bands are almost the same as you see here, then that's a good loading control. Every time when we are analyzing a Western blot result, we need to use these loading controls values, and there should be a subtraction or a division from the expression of the proteins that you are looking for. So now let's jump into the analysis. But first you need to select the bands. I assume that, for example, here, it would be easy to see that the first and the second has a high spot on it, or like the band is really thick. However, the third and the fourth are a bit different. Here also there is a difference. So with the densitometric analysis, the aim is to validate the difference what you see on the figure. So in my case, I'm going to do an analysis on this protein 3 and the loading control. There are 2 types of person in the lab. The first one who is going to go by one selection and analysis, and the second who is going to use the whole line of bands and do the analysis. The difference between them, that one of them are going to use a lot of mistakes during the analysis, and I'm going to show you why. So if you go with the single selection, which means that these rectangular ones, with the left click, you can select the band that you want to see. If you are going to do the selection, it is important to take care of the size of this selection because the band should fit inside perfectly. And after you selected it, there is a button combination, which means like Control 1, and then or one sign, a number will appear inside this selection. So that shows you that you have selected one of the bands. With the cursor, you can easily move to the right, and that's how you are going to select the next one. So here again, you need to focus on that the band should fit inside this kind of selection every time, and do not care or worry about that if it's overlapping with another one. It is not going to matter at the end of the analysis. But because this is the second band of yours, you need to push the Ctrl 2. You need to repeat this step one by one until you reach the end of your figure, for example, here. So I'm going further, I'm going to move the cursor to the right until I reach the third one. But here, because this is also another lane, you need to push the Ctrl 2 again. But as you see now, this is the third lane I'm just working with. And I'm going further with the fourth one. Now I need to push the Ctrl 2 again. And now it's time to reveal the, how can I say, the darkness level inside these squares that we selected. So if you push Ctrl 3 with the Analyze, and then you click on the jails, if you see, the Ctrl 3 will be the plot the lanes. So you can reach this one here as well if you want. If you click on it, then the program will draw you the curves. And the major point is that during these curves, you need to draw a baseline, and also you need to calculate the integrated density under the curve. But the problem with this one, if you have a selection, as you see here, it would be easy to, let's say, select where was the baseline. But here, it is not so easy. So in that case, whenever you are using the baseline correction, which the program automatically gives you these straight segmented freehand lines and arrows, you need to select one which fits almost perfectly on this one. But let's imagine you go further and you just doing something like this. It is not going to work. And also here. Or if you scroll down, oh, that would be easy, right? That's amazing. But the problem with this one, that as many lines you are using, as many mistakes you are going to generate. So in that case, that is not really efficient and accurate regarding to the analysis. So I'm going to show you another one which works more easy, I think, and the beneficial part is that only once you are going to make a mistake. So I'm going to close this one, I'm not going to save it, and then I'm going to click on the rectangular selection again. Because my figure is ready, I'm going to do a selection which goes in a way that I'm going to select all the lanes. Yes, inside now you see 1, 2, 3, 4, 5, 6, 7, 8 bands are there. So the whole lane is going to select it, and then I'm going to push the Ctrl 1. Usually the program is going to ask you, are you sure that this is horizontal? Because I sense that there is a kind of bending on it. But yes, I'm pretty sure that my lanes were selected properly. So this is the first selection, and then I go with the cursors to the next one. For example, look at this. Now I'm going to push Ctrl 2. And then I'm going to make a selection and plotting what I have selected. So I'm going to push Ctrl 3. And now I had the results in a picture like this. I think now it's more easy to select the baseline because then it should cut all these curves. So in my case, I'm going to set the baseline, which should be set like this. And also I'm going to do the same thing on the lower part, but this lower part was the second of mine. So I'm going to, as you see, that was the first one, which refers to this one. This peak is referring to this one. This is the third one, and this is the fourth one. So I'm going to set everything to the fourth one and to here. So basically, I need to cut my gels like this. But if you see, this is not going to work because then I'm going to cut everything or most of the peak of the last one. So what I do now, I'm going a bit lower and then I'm going to use the lane, which should be straight. I know it's hard to find the straight one, but try to be as precise as you can. So now the question comes, how you are going to generate data from this one? It's easy. You just need to separate the peaks. And the peak separation can work with the same feature of this program, which means that you need to cut these into the four or like the other set of samples. What does it mean? For example, here in the case of the gap DH, as you see, that was the first peak, and this was the second one, and the third one, and the fourth one. So you need to close these peaks with straight lines. And this is how we close the peaks. Now you can ask like, but hey, what's going on with the overlapping features? Because if you see, there should be here some overlapping and there overlapping. In this case, that is not not part of the analysis which people call like, oh, this is manipulation or fabrication. These lines are theoretically closing the marks of the dense black parts. So these peak and these ones, for example, they are showing you that you had some black dot on the figure. So that's why this place here, which was not dark enough, you can close it with a single line. Also, you need to close the analysis at the end, otherwise you would get a really weird value. In the other case, when you're going to work with this one, you need to do the same thing. So you just need to cut the bands and in that sense the peaks. Here it is important to have the straight lines, otherwise there is the middle of the error, I would say. And if you have closed everything, now you need to get the data. So in here, if you had like 8 samples and also 8 samples here, it works to check the figure. So in the case of the figure, if you see you had 1, 2, 3, 4, 1, 2, 3, 4, so 8 samples are there. And these peaks and the dot size should refer to each other, which means that if this is high, that should be high as well. So now let's see which data you would get. For that, you need to do the analysis, but that goes with the magic wand here again. This is a tracing tool. If you click with the magic wand inside or under the curve area, then the program is going to select it and mark it as yellow, and then it gives you the value immediately. So in my case, this is the first one, the second, the third, and the fourth, 6, 7, and so on and so on. So here, now the program showed me the values. So take a look. The first one was 12,000 something, 13,000, 11,000, and 8,000. Okay, let's go further. 12, 14, 15, and 13,000. And that was for the gap DH. So now, or the loading control, sorry. Let's take a look at this one. I'm going to go with this one, and 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 so on, and so on. So basically, now you have 16 values, and these values needs to refer to the picture. I'm going to close the analyzers. Don't save it. And then I'm going to take a look at the values. These are the ones that you need to use for the analysis. Later on, you can select all of them, copy it, and place it into an Excel file. If you place it into an Excel file, you need to corrigate the values, even if you're using a Hungarian setup. We had two analyses here, which was the loading control, and also we had the protein 3 expression. So now I'm going to use the proper selection, which means that I had 8 samples. No, it's not going to work like this. Is how it's going to work. And the mechanism, or like the analysis, how can I say, the strategy, most of the Western blot analysis is that we need to divide the protein expression with the loading control. So I'm going to do the same thing. I'm going to use the protein values, and I'm going to divide with the loading control one. As you see at my computer, it is not going to calculate because I have a Hungarian one and it is working with the English one. So I need to change all the points to commas. And for that, I'm going to use the Control H. I will put the points there, and I'm going to change into commas. I'm going to change all of them. And now I have value, right? Let's see. So this is a calculated value. And if you want to show this as a graph, that's how we make a figure like this. This is how it would look like. So now, before you go for the further analysis, it's really important to check whether you did a good job or not. So I'm going to use now the picture I was using for calculation. So I was using the protein 3 expression and the loading control. So now let's see. The first one was high, that's correct. The second one was, let's say, high, but based on this figure, I won't see a big difference. But the third and the fourth would be lower and lower, that's for sure. The same thing happens here. It is high and this is also high, but compared to these two, it was a really, really low expression. So I would say that that's a good type of analysis what we have done. But there are some cases when you make a mistake. I'm going to show you how it works. For example, here, if you had two selection, and also if you click on the jails and plot the lanes, now I need to redone the whole thing. Let's see this one, the control 1, and this one, and I'm going to plot it. So I'm going to delete the data for the previous one, and then I'm going to make a selection now. For example, I'm going to cut the story here, and now I'm just going further and further. But if you take a look at this figure, it looks like that I closed almost everything, right? And then I'm going to use my magic wand to do the integration. I'm going to click. Wait, why is it so yellow everywhere? That happens, which means that you did not close properly the area that you want to use as a calculation. Here it is okay, and here as well. and here okay, and that's fine. But here again, so now I get only just 6 values. So if you would realize during the analysis that you get an outstandingly high value during the analysis, might be the case that you did not close it properly. So please take care of the yellow part which shows you the selection, because otherwise it is not going to work. And or it is going to work, but you would get a false data at the end. So I would strongly recommend you to be sure that you closed everything. For example, here at the end, and as you see here, it almost looks like that it is closed. But if I just make a magnification on the figure, it is not closed, as you see here. So that's the thing. You need to take care of the values, because if you made a wrong selection, then your calculation in the Excel, it will be also different. So it's worth to put the figure back to together with the data and just check it whether it is referring to the figure or not. So this is how we do usually the analysis of the Western blot with the image G, and as many repetition you have, as many times you need to do this kind of analysis. And then from the calculated values, you can make a mean calculation, and also the graph is going to do instead of you. But the question comes, what you are going to put on the title? Because what you did, it's a densitometric analysis, but here you were using optical density. And because you will do an integration, the one that you need to type on the axis should be the so-called integrated optical density. Let's see, that should be black. So that should be this integrated, I made a mistake here, yeah. So integrated optical density, that should be the title. And here you can type the name of the protein that you were looking for.